All right, we're going to talk about DNA sequencing. When we say DNA sequencing, it refers to methods and technologies that are used basically to determine the sequence of DNA. And when we say sequence, we want to know the orders of nucleotides bases, which are going to be adenine, thymine, guanine, and cytosine. So when we talk about DNA sequencing, these are going to be methods and technologies that we're going to use. There's different ones. Gel electrophoresis, there's going to be STR analysis. There's a lot of different DNA sequence methods. Now we're going to talk about the reasons we would use this. DNA sequencing is a key technique used in nearly all molecular biology labs. So when they want to look at certain molecules, if you were studying molecules in great detail, that's the field of molecular biology. You would want to know the DNA sequence of different molecules, not molecules per se, but you know, maybe for a plant, if trying to figure out something. It's used in genetics labs to help you basically know more about genetic conditions. It's used in forensic science to help, you know, catch killers. So if you were wanting to go in forensic science, you'd be looking a lot at DNA technology. It's also used in bioinformatics. Whoops, I misspelled that. And even a lot of labs that want to study evolution would use DNA sequencing. There's so many different ways that we use DNA sequencing. So I'm just going to put etc. Now, one of the first sequence methods that we ever had, and this is the one I'm going to teach about, is called the Sanger sequence method. So I'm going to put one of. This one was one of the first DNA sequence methods was discovered in the 70s. The scientist's name is Frederick Sanger. I guess he went by the name Fred. So the first DNA sequence method was called the Sanger, for his last name, sequence. Now this was basically the breaking edge technology in the 1970s. It was the most used for a very long time, and how we came up with a lot of our other DNA sequencing methods. So I'm going to teach about the Sanger sequence. And when we talk about the Sanger sequence, what Sanger did, Mr. Sanger, is he took a DNA. So you have to remember that DNA is a double helix, and facing on the inside are nitrogen bases, and we always do these in sets of 3 called codons. Now, in order to read a foreign DNA sequence, it had to be unzipped. So, what we call the replication fork. So we have 3, 3, 3. Basically, that would continue. So what they did is they used helicase to unzip this molecule, and they put this DNA into a chemical reaction, and they had to do a couple of things. So we're going to take this and we're going to call it the template strand. And the template strand in replication is going to be used as a way to copy. So in the Sanger sequence, we take this strand of the DNA and we figure out the template. So we took half of the DNA. Let's just say it's ATTGCC-CTA. This is the template strand. Now we're gonna put this into the Sanger sequence process, and what we have to do is we have to basically take radioactive compounds and attach it to each one of these nitrogen bases. So in the Sanger sequence, what we do is we're going to use DNA polymerase for 4 different chemical reactions. So if you remember, DNA polymerase is going to be the enzyme that makes new DNA. So to figure out the sequence, we have to use 4 DNA polymerase enzymes, 1 for each base. So we have a DNA polymerase for adenine, and we're going to have a DNA polymerase that's going to make only thymine. 1 DNA polymerase is going to make only guanine, and 1 DNA polymerase is only going to make cytosine. So we're going to have 4 chemical reactions. Now, another thing during this process is we have to radioactively attach or label each of our nitrogen bases. So we use radioactive or isotopes. So we radioactively attach phosphorus and sulfur to adenine so that it basically becomes colorful or fluorescent and it glows. Any thymine that you have, we're going to make it radioactive, and I'm just going to say that it's going to glow orange. Cytosine is going to be radioactively labeled as well. So we have a DNA polymerase for each one of these. We have adenine over here, which is radioactive blue, and we're going to make these glow. We're going to use radioactive labeled isotopes. Now I need to grab another color for guanine. So these each are going to have their own radioactive compound attached to them. So the DNA polymerase is going to do 4 chemical reactions. So this is the Sanger sequence. 4 chemical reactions. So adenine is going to basically come in this chemical reaction with its own separate DNA polymerase. Thymine, and these are glowing because they're radioactively labeled, guanine, and cytosine. So basically, we have 4 chemical reactions, 1 for each nitrogen base, which are radioactively labeled. So you have the first chemical reaction, the second chemical reaction, the third chemical reaction, and the 4th chemical reaction. Now we can put on here for the Sanger sequence, this is a Sanger sequence. We can say that these are radioactively labeled. So basically, we can take our DNA strand and we have 4 chemical reactions, and then they're gonna come up into what we call 4 columns. So your DNA fragments generated are gonna be separated in a gel. So this is basically a gel. So your DNA fragments for adenine are gonna be in this first gel column, and then you have your thymine, the second gel column, your guanine, which is your third gel column. So each one of these, adenine, thymine, guanine, and cytosine, are going to basically be different fragments that are generated by running the DNA sequence, and we have to make copies of it. So the reactions are going to be separated. So all of your nitrogen bases are separated by gel electrophoresis. So we have 4 columns, one for adenine, one for thymine, one for guanine, and one for cytosine. These are all different colors because they're radioactively labeled. Now, after this process occurs, something else has to happen. So for the Sanger sequence, in order to read it, they had to take paper, or an X-ray, which I'm going to use as a paper. Say this is an X-ray. And they had to expose the X-ray, which could be anything. I'm trying to find something that I can use. So when you do an X-ray, you have X-ray film, which is in a box. And we'll use my binder here. So you place your X-ray film in your box, and basically you're going to expose it to these radioactive elements. So you have your X-ray film inside of your X-ray box. You expose the radioactive elements or nitrogen bases to your X-ray film, and you're going to process your X-ray film. And when you process it, you end up with a picture that looks like this. So it's no longer color-coded. This would be basically the X-ray. It's been exposed. So at the top, you see the 4 chemical reactions, you see the 4 columns, and this is the Sanger sequence. So when we look at this, the radioactive DNA fragments show up on the X-ray film, and we know that each one is a nitrogen base. So this right here, DTATP, these are going to correspond to your nitrogen bases. So your nitrogen base adenine, this one was adenine. Anything that falls in this column is going to be adenine. Then you have thymine, and it's just got DDT-TP, so that's thymine. The next one is guanine, DDG-TP. Our guanine was green. And anything in this column is guanine. And then you have your cytosine. Your cytosine is in your last column, and it created this. So now this is the X-ray film, so it's not going to be glowing. It's not radioactive anymore. But you can use the Sanger sequence to basically figure out the DNA sequence from which you ran. Now, it's only going to be a copy of the original sequence. So you're running the template strand, which could be this, it can be anything. You run this through the machine, and it's doing the copies because DNA polymerase would basically go through, and it would say T, this one's A, C, G, 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 A, T. So your DNA polymerase is basically figuring out what the template strand is. So this is not the template strand, this is the Sanger sequence, it's the copy. So in order to determine the DNA nucleotide sequence of the DNA, this foreign DNA, you always start on the bottom right here. So you have to read the Sanger sequence. So the Sanger sequence always starts like this. You start on the bottom. You would start at the very bottom. Now you're going to read it all the way across. Since adenine is on the bottom, and we know it's adenine because it's in this column, and it's at the very bottom. So you start at the bottom. Now you could have one that's over here. Whichever's the lowest one is your first one. So your Sanger sequence starts at the very bottom, and it's always going to be in groups of 3. So you can do this either 2 ways. You can say all of these are adenine, but you're going to do them in groups of 3. So you can go 1, 2, 3, and then you do your first 3. So you can just say 1, 2, 3, 1, 2, 3, or you can keep going. So your Sanger sequence would be adenine, thymine is the second one. So starting from the bottom, that one's the next lowest one. This is the next lowest one. So this one falls under guanine. So then you draw a little line because DNA is always read in sets of 3. So you go to the next lowest one, which falls under guanine. 1, 2, 3. I like to do mine in sets of 3. So you have 2 guanine, so it'd be G, G, and then over here, cytosine. That's 1, 2, 3. Now you go up. You draw another line for your codon sequence. So 1, then it comes over here, 2, the next lowest one, and 3. So you just follow it up and you say the next Sanger sequence is cytosine. Then you say thymine. The next one is adenine. Now you continue to do this all the way until the top. Now, this is the Sanger sequence. This is only a copy of your original strand. So we use the Sanger sequence to determine the nucleotide sequence of the original DNA. So we took our original DNA and we basically made a copy. So the new copy that you're making is going to be the Sanger sequence. That's what this is. So to figure out the original DNA, you're going to go opposite of the Sanger sequence. So the original DNA would be T, A, C, 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 G, G, A, T. So this is the original template DNA that basically we discovered. So we can figure out what DNA is. So if we were using this in forensics, if someone murdered me and they left their DNA behind and all we had was Sanger sequence, we could figure out the murderer's DNA sequence by using Sanger sequence by the ways that I described. Now, since we know that Sanger sequence figures out the original DNA, as scientists, we can also figure out which proteins are going to be made using the Sanger sequence. So we go through the central dogma. We use the original DNA. So we're going to use the Sanger sequence. So we're going to write this out. The Sanger sequence I'm going to write it on another paper, is A, T, G, 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 C, C, T, A. This is only a copy of the original DNA. So then our original DNA, you had to figure it out using the Sanger sequence. So our DNA strand is TAC, CCG, GAT. Now, we can use the DNA sequence that we figured out to figure out what genes it encodes. So the central dogma is when you go from DNA to mRNA, that is called transcription. And then when you use mRNA, you can figure out your amino acid chains, which is going to basically be a protein, which is called a polypeptide, which is amino acids held together to basically make your protein. So your amino acid sequence. So in lab, you're basically going to use your Sanger sequence to figure out your DNA. Now, the next step in lab, and it's a question on there, it says figure out the Sanger sequence, then it says to figure out the DNA. The next question is to determine the mRNA by matching the nitrogen bases to your sequence. So I'm giving my original example here. Y'all's is different. So you have your Sanger sequence. Now you have your DNA. The third thing is asking you to do transcription. So transcription is going from DNA to mRNA. So number 3, you're going to take the DNA sequence and you're going to convert it into mRNA. Now, the only difference with mRNA is that you never have thymine. So when you look at your DNA, you can figure out your mRNA. So opposite of T would be A. You can have A's, you just can't have T's. So the opposite of A is U. So then you go here. So I'm using the DNA to figure out my mRNA. Now these are also always read in sets of 3. So AUG, G, G, C. The next one would be C, U, A. Make sure you do a mental check when you go through here and make sure that you have no T's. Thymine is never found in RNA. Now, Sanger sequence, yes. DNA, yes. But in step 3, you're taking your DNA and you're figuring out your mRNA. Now, the next part of this is going to be using the genetic code. The genetic code says that we basically always read DNA in 3 codons, and then we make mRNA, which is read in 3 codons, and we can use mRNA, which is going to bind to your ribosome, and we're going to make the amino acid sequence. So that is called translation. I'm looking for a piece of paper that I need to show you. Of course, I lost it. In your lab, you're going to have a piece or a chart that is the genetic code. The genetic code is going to be used to help you figure out your RNA amino acid sequence. So you use your mRNA. I have one somewhere, I just have to find it. I can't find it, but that's okay. I can kind of draw it up. Now, to do step 4, to figure out your mRNA, to figure out your amino acid sequence, you're going to use a chart. The chart is called the genetic code. This was basically described in lecture. So if you watch those videos, you'll have seen the chart before, but we're going to take mRNA from step 3 and we're going to use the chart. Now the chart is written out something like this, and it's got the first base over here. This is how you read it. Let's just say it's A, U, G, C, and I could be getting this wrong. The second base over here would be A, U, G, C, and then your third base over here is going to be a little bit smaller. It's AUGC, AUGC, because you can have multiple bases. AUGC. It's something like that. So when you read your mRNA, this is your first base. So you would go A is your first base, and then the second base is AU. So this would be all listed as AU, and then the third base A, AU, G, AU, C, and AUU. I missed AUU. So in this, you're going to have basically 3 things. Now these are called your codons for mRNA, the codes. So this is called the genetic code. The genetic code is giving you 3 bases. When you have 3 bases like AUG, that is called a codon. So the genetic code is listed, and then out to the side, it's going to have an amino acid that goes with it. So AUG is always going to be methionine, MET. So this over here is the amino acid sequence. So methionine is the amino acid that goes with AUG. So for step number 4, you would say AUG, you'd use the genetic code and you'd find AUG, you find the code AUG, and then you say that the amino acid would be met. So step number 4 is you would say the first amino acid is MET. So this is number 4, MET, and you still put a line. I like to circle this to represent it's my amino acid. This is now a peptide bond. So you would go to your next code, GGC. So you use your first base here, G, going across, second base, G, G, and then it would be C. So here's your codon, and out to the side, it's gonna also have an amino acid out to it. Now that amino acid could be leucine, it can be arginine, it can be anything. And I don't have this chart memorized. You'll never have to memorize this chart, but you will have to take a Sanger sequence, find the Sanger sequence, then do the DNA sequence, then do your mRNA, and then figure out your amino acid sequence, which is going to be translation. So you're going to have an amino acid, and then you're going to have another amino acid. So that is how you do the Sanger sequence. I'm sorry I lost my paper, but I think that you should be able to do that. There will be one of these on your post lab. There will be one on your quiz, and there will be one on your lab practical. Now on the lab practical, each of these questions is 4 different questions. So you do need to know how to read a Sanger sequence, and it's 4 questions. You figure out the sequence, you figure out the DNA, you figure out the mRNA, and you figure out your amino acid sequence. So make sure that you understand how to use this whole thing.